Imagine millions of short nucleotide sequences pouring out of a sequencer, demanding to be quantified and categorized at high speed. You map these raw RNA seq reads to a reference genome, quantify the transcripts, and run the counts through an algorithm to see what is over or underexpressed. This left-to-right flowchart shows a smooth progression, from raw reads through salmon and into DSEC2, until the process hits a wall. You have a list of significant features labeled with machine-readable ensemble IDs. You must convert these into human-readable gene symbols, but this simple step splits into multiple points of failure: broken web addresses, outdated gene versions, or dropped symbols from mismatched reference tables. If the mapping step drops 10% of your data or mismatches an isoform, the downstream pathway analysis carries that error forward. Inconsistent annotation doesn't just lose data, it introduces noise that can obscure the biological signal you're trying to find. If you're pulling a quick CSV file of top hits for an afternoon lab meeting, a few dropped gene names won't ruin your day. But compare that to combining 5 different clinical data sets for a multi-omics meta-analysis on FSHD, targeting a high-impact journal. You have to decide how to build your pipeline. You can use the quick, legacy lookup functions scattered across older coding tutorials, or you can invest the time to build a rigorous bioconductor architecture. If you use an unstable method, you face a serious problem during peer review. 3 years from now, a reviewer will ask you to rerun the code. If your script pulls from an updated database, your exact p-values and gene lists will change, and your original results will be lost. The annotation tool you integrate on day 1 dictates the lifespan, auditability, and scientific validity of the entire project. Let's look at the first option, legacy lookups. This includes manually uploading text files to a web browser or loading static R data packages like annotables. This comparison matrix evaluates tools on setup effort, network reliance, and versioning rigor. For legacy lookups, setup effort is 0. Feed it IDs, get gene symbols instantly. However, older static packages lock to outdated gene models, and web uploads leave no programmatic trail for reviewers. Prioritizing immediate speed by clicking through a web portal removes the code trace, making reproducible standards impossible to meet. The second option is the live database query, most commonly executed through the popular BioMart package. BioMart requires building a specific R query that dynamically pings ensemble servers. Because this relies on an active network, server outages cause immediate script failure. Even worse, rerunning queries later pulls updated gene models, silently changing your output without warning. While dynamic queries give you flexibility, relying on shifting external servers means your pipeline cannot be trusted for a longitudinal study. That brings us to the 2026 standard, the modern hub stack. This relies on Annotation Hub, EnsembleDB, and TIX Meta. This modern stack has a steeper learning curve, requiring more verbose code to leverage Bioconductor's cloud caching. But instead of pinging servers, it downloads a version-locked ensemble database locally. TIX Meta then automatically attaches the exact genome build and provenance directly to your data. By embedding the correct annotation data early, complex downstream steps like gene set enrichment through Cluster Profiler run smoothly, accurately cross-referencing between transcript lists and known biological pathways. The extra lines of code you write on day 1 are the exact cost of securing an audit-proof, locally stable infrastructure. This quadrant chart maps our 3 tool families across the reality of a production environment, plotting upfront effort against long-term reproducibility. Legacy packages and web tools sit entirely in the bottom left. They take seconds to use, but fail basic audit checks. BioMart sits directly in the center. It requires coding knowledge and leaves a script trace, but remains too vulnerable to network shifts to meet strict publication standards. The modern stock moves to the top right quadrant, demanding heavy upfront configuration to guarantee absolute data stability. Committing to that top right quadrant means you are actively choosing to protect your own time and sanity months down the line. There is a direct trade-off between the immediate convenience of a lookup tool and the long-term stability of the analysis it generates. If you were just running rapid, exploratory data analysis to see if an experiment worked and you will delete the output folder by Friday, using the bottom left quadrant is perfectly fine. But if you are building a meta-analysis or finalizing the primary data for your PhD thesis, moving to the top right quadrant is the standard for work intended to survive peer review and long-term reuse. Spending 1 extra hour today to learn Team Meta and cache your ensemble database locally saves you 2 solid weeks of frantic, panicked debugging during peer review revisions next year. Researchers must stop relying on live web queries for any code intended for publication. True bioinformatics engineering is not about writing the shortest script. It is about matching the rigidity of your pipeline to the